Using alcohol wipes before placing electrodes ensures there is a sufficient electrical signal.
Electrodes must be placed in the gap between the second and the third rib to measure the amount of electrical supply to the respiratory muscles and on the collarbone for reference.
Electrodes measure the amount of electrical supply to respiratory muscles.
The equipment must be calibrated for accurate results. The red and the blue lines are recording the heart and the respiratory muscles, whilst the green line is measuring the airflow in and out of the mouth.
Plasticine is placed around the mask if it didn't fit the subject, as air escaping the mask would generate an invalid result.
Subjects must rate their experience on a scale in terms of tension and relaxation depending on the condition.
So I got a studentship from the Physiological Society. They awarded it to us, so we wrote in an abstract and a bit of background research about what we were going to do in the lab. And about 100 people applied, and they gave out just under 40 awards. We're using EMG para in our study, and it measures the activity of parasternal intercostal muscles. Which means you stick 2 electrodes on the second intercostal space, so about here, and then 1 on your collarbone as a reference, and then you stick electrodes to them. It's been used a lot in research in populations where they can't perform more complex functions, such as spirometry or max manoeuvres. For example, if they're a child, or they're in intensive care, or they're wheezy. And research has shown that a period of acclimatisation can change the measurements for EMG para. So the longer you're in the lab, the more relaxed you are, and you get slightly different measurements to when you first come in. And often in order to relax participants, they play music or they put videos on to distract them. And what isn't known at the moment is the effect, if there is an effect, of the different stimuli on EMG para activity. So the aim of the study is to look at the effect of different stimuli. So we have a tense video condition, a calming video condition, a music condition, and one with no stimuli, and to measure the effect of different stimuli on EMG para activity and respiratory rate and tidal volume. So I was in the student panel 3.5 years ago. That was when I was at JFS, and I'm now going into my final year studying experimental psychology at Oxford. So this project is really exciting because it combines aspects of psychology, so the different stimuli, which is to do with my degree, and then physiological measurements. In my degree I do psychology, neurophysiology and neuroscience, so it's just combining a lot of different areas, so it's an interdisciplinary project.